Hi everyone, welcome to the July Recon Community Meeting. Today we have some exciting talks by two of our speakers, Manu Lera Ramirez and Bijoy Desai. And we'll start with Manu, who is a research fellow at the University College of London. And he did his undergrad in biotechnology at the Universidad de Zaragoza, I hope I'm pronouncing that right, in Spain. And he followed this up with a master's degree in biochemistry at the University of Geneva. And he went on to do his PhD at Heidelberg in biophysics, looking at fission use. And also, and then a postdoc at the Institute Curie. And now he is a Maria Skłodowska-Curie Research Fellow based at the Biolab at the University College London in the UK. And for his work now, he's been developing a tool called OpenCloning, which is an open source web application that's useful for planning and documenting genetic cloning, genetic engineering, and doing different cloning strategies. So we are excited to invite him to tell us more about this tool, OpenCloning, and hopefully you will be interested in using this and giving him some good feedback. So I'm going to pass it over to you, Manu.
Thank you, Yankai, and thanks for inviting me. Really appreciate it. So I'm just gonna put my presentation so you all can see it, I hope. Okay, I see nodding. Good. So yes, so as Yankai said, my name is Manuel or Manu, and I'm a postdoc in UCL in London, and I'm going to talk about my postdoc project, which is called OpenCloning. And OpenCloning is an open source web application to plan and document cloning. And I always start with this kind of cartoon in which I depict, some DNA in a kind of cave painting way. And the idea is that we have very rudimentary ways right now to capture how we perform cloning in the lab. So generally we do it as text. And can we move to a more systematic description that also enables automation? So that's kind of the project. So, I mean, if you are in this seminar, probably you don't really need to be taught about this, but just in case, we when we talk about like provenance of sequences or the lineage of a plasmid, we're thinking of what were the steps, the manipulations that we follow to go from existing sequences to the particular sequence we have right now. So, there's different ways of doing this. One of them would be using restriction and ligation, like I'm showing in this example, where you would use restriction enzymes to cut bits of a sequence and then ligate them to form a new one. But as you know, there's many ways to do this. And starting from the same inputs, we could produce the final plasmid in different ways. But it all comes down in some way to cutting and pasting sequences. But despite this being such a common thing, we still don't have an open standard to encode these informations. We don't have a kind of a data model or a file to describe deterministically these steps that we follow to go from existing sequences to new ones. So I tried to summarize the situation with this meme. So I leave you like 10 seconds to have a look at it. So the idea is we do have some tools out there. So if we look at what people are using in labs, if your lab is cloning, there's a good chance that they're using something like Benchling or Genius or Snapgene. And these tools will allow you to plan your manipulation, your cloning. But for example, Benchling will not always store the history of what you did. And Snapgene and Genius will do so, but they will do so in a proprietary format that you cannot use in a meaningful way outside of this software. So you can access the history of something in Snapgene or Genius, but then you cannot use this history in a script. You cannot integrate it with your systems or anything like that. And this is kind of like an open science aspect of it, which maybe you are interested in. But even if you don't care about open science, even if you don't care about like sharing or like open source, all this kind of stuff, this really matters. And the reason is because we don't have a standard to represent these things. We cannot automate things, right? You don't have a kind of description for things. You cannot write code that automates the design of DNA and all that. And that's why these programs you can only use through a user interface. So you have to kind of click through the system because they don't have a way for you to automate it. And there are some like academic software that you can use. So for example, Ape is kind of a bit legacy software, I would say, but you can still use it for cloning. And there's also Python libraries such as PyDNA, which is something I use very often. But still, there is no way to kind of capture this cloning history. So that's what OpenCloning is about. So the goal of the project is to produce a data model to represent the provenance of engineered sequences, but at the same time also develop a web application with an intuitive user interface to work with it. So the idea is that, okay, we have a great open data model to represent cloning, but not at the expense of a good user experience. So you are still able to kind of like click through an interface and produce your cloning without needing to know how to code or how to interact with these data formats that maybe not everyone is comfortable with working. So very briefly, the idea of the data model is that every sequence comes from a source, and this source may be an external import. So a sequence that we imported from a file, or a genomic sequence that we access from the NCBI, or a plasmid that we have got from AdGene. So basically something that comes from somewhere else and we read into our cloning. Or it can also be, a source can be a manipulation, a step that takes some inputs and produces an output. So for example, a ligation like in the example above, would be the source of a new sequence that takes some other sequences as an input. So then you can see that through chaining sources one after the other, we can reconstruct the history of some sequence and see where it came from. So that's it. That's all I'm going to say. And now I'm going to show you a little bit how it looks and how you could use it on your everyday work. So you can go to opencloning.org and you'll find the tool that I'm going to show you now. So when you go there, you'll see something like this. So we start from some kind of clean slates. And now we're going to start importing sequences. So one way to do that is, for example, from AdGene. So if you are using plasmid from AdGene, you can go here, you can click on repository ID, you can select AdGene as a repository. And then if you provide the identifier of the plasmid from AdGene and you click on submit, this will make a request to AdGene and give you your plasmid. And you can see it here in a small map, but you can click on the eye and it will take you to a sequence editor, much like the SnapGenes, benchlinks, and so on. So you can kind of like see the sequence, select bits, etc., etc. And another way to import sequences that I think is quite useful and very much like different from what you have in SnapGene and benchling is that you can directly access genomic sequences. So say that you're working with coli and you want to access the sequence of a particular gene in the genome. You can do it very quickly with OpenCloning. So you come here, you go to genome region, you select a locus in the reference genome. But if you are using a different assembly, you can also provide the assembly identifier. And then we're going to select E. coli. And then let's say we're going to get laxeds. And then you can select upstream and downstream bases from the gene. So we can, by now, the default is 1000. And then we make a request to the NCBI, and we get the sequence of the gene in here. So, and then once we are here, we can start doing cloning. So we can click on this plus sign and then use this as an input for a new kind of manipulation. And for the example, I'm just going to do homologous recombination. So that's typically what I was doing when I was working in yeast. So what we would do back then would be, okay, if we want to delete this gene, for example, what we will do is we amplify these cassettes, this resistance cassette with a PCR, and then we will add some homology to the primers, so some tails to have homology and replace something in the genome. So let's imagine that's what we're going to do. And for that, you can say, okay, I want to use this sequence. I want to use it for a PCR. And here you can design primers. In this case, you can design them for homologous recombination, and then you can specify that your target sequence is this one, which is the genome, and then you click on design primers, and this will take us to a interface where we have to select what do we want to amplify from the plasmid. So in this case, we would want to amplify a resistance cassette, but you can select any arbitrary region. So let's say the kanamycin resistance cassettes. What do we want to replace in the genome? In this case, for example, the LACZ gene. And then you can specify how much homology do you want, what TM do you want for your primers, etc., etc. And then when you click on design primers, it will propose 2 primers that it has designed. And if you're happy with them, you can save them. It will put them in this PCR. Then you can do the PCR and finally do a homologous recombination with your PCR product and the genome, and you will get the final sequence of your allele that you've made in your strain with it. And then you can also, for example, if you have sequencing data, you can click in here and you can align your sequencing data with the sequence that you have generated here. Then you can also export this sequence or this file. So you can export this as a GenBank or FASTA file, your final sequence, but you can also export the entire thing, the entire cloning history as a single file. So you can click here on File, Save Cloning History to File, and you just give it a name, and this will download it to your computer. And for example, you can send it to someone else, or you can keep it for your records. And if you load it again on the site, it will basically show you again everything you did. And if you had, for example, sequencing data, it will also have the sequencing data there as well. So let me just see if I'm missing something from my scripts. Yes. So something else that you can do with it is you can also use something called Planotate. I don't know if you're familiar with this tool, but it's basically a tool that automatically finds features on your sequence. So for example, I have an example in the interface where I provide a naked circular FASTA file. So I have just a sequence without any feature. And then I run Planotate on it, and it finds all these features in it. So I could run it again, but it takes like 15, 20 seconds. But basically what you would do is you come here, you take your naked one, you click on annotate features with Planotate, and then it will make a request, it will take some time, and it will spit out your annotated plasmid. And finally, one more thing I wanted to show you is that you can also make, you see, it annotated the plasmid, is that you can make templates. So for example, if you have a cloning kit, so for example, like the reclone distribution that is, you know, it has a Golden Gates syntax and there's a certain way it is meant to be used, you can make a template. So here there's a handful of examples for cloning kits that are available from Adgene as Moclo kits. So here I'm just going to load an example for a Moclo kit. So as you can see, you have the different kind of components of the assembly, and then you can select from a drop-down menu each of them, or provide your own if your plasmid of interest that you're going to put in there is not part of the kind of standard kit. So if you're like distributing plasmids, or if you want to like share your methods, you can also think of making a template like this to help people clone using your method. And finally, I think the other thing I wanted to show you is that everything I've shown you here on the website, you can do with a script, because everything is in the backend working in Python. So all the operations that you've seen, the primary design, the cloning, the making the history that then can be loaded in the website, all of that you can do in your writing a script. And then you can think of some more advanced use cases. So I recently did this template for a collaborator in the University of Bordeaux. So basically, they had developed these cloning method to do gene deletion in Arabidopsis, and the idea was that they would clone a bunch of gRNAs on a single plasmid. And the way they would make them would be by putting, oops, sorry, that's my bad. They would basically put these gRNAs as spacers in primers, and they would amplify many times a tRNA motif that is cut inside cells. So basically, you end up with gRNA, this motif, gRNA, this motif, gRNA, this motif, and when this is made into RNA, the cell will cut between the RNAs and release them for the CRISPR to target the different targets. So it's quite a long cloning strategy, which involves first doing these PCRs, then you do Golden Gate, and then you do 2 midways to produce your final plasmid. But in the end, the only kind of user input that you're giving is those 4 gRNAs. All the rest is checking whether the overhangs are okay, they are not clashing with other overhangs that are part of the constant bits. So you can make a form like this in which the user only provides the minimal information. So in this case, 4 gRNAs. And then when you run the form, it downloads a file that contains the complete cloning strategy. So if you then load this in the interface, then you can see the full cloning strategy that you would have to follow. And you can see that there's a lot of steps, and there would have been a lot of things where you could have made a mistake. And with this, you would know. So I think this is another aspect that can really help, like to minimize the user input that you need to produce a full-on cloning strategy. And also, if you have to do it 6 times, and you're doing this in Snapgene, it's going to take you forever to like click through the interface one time and over again. So yeah, so that's, I think, everything I wanted to show. I think that's pretty much all of it. Yeah. And so, yeah, I mean, the project right now is what I would call, it's in a situation where I would call arcade mode. So you can basically load files, you can download files, but there is not a database where you persist your sequences and save them. But this is coming. So in the future, there will be a way to save your collection of plasmids, strains, and all that, and see what comes from what within the relationships. And in the meantime, I'm also doing integration with electronic lab notebooks so that you can, if you are using an electronic lab notebook in which you have a database, you can read sequences from there and clone using OpenCloning and then save to your electronic lab notebook. And I already built an integration with eLab FTW, which is an open source electronic lab notebook. And this is already live and it's being used in some universities. So that's it. So my final slide is, okay, you've heard all of this. Why would you try OpenCloning when you already are using something else? So I think what it shines is it has better support for genome regions. As you saw, you can query by gene and get this very quickly, the sequences from the NCBI. It already has more methods than Snapgene and Benchling. It has everything that Snapgene has, plus homologous recombination, plus CRISPR, and also Crelox, which is not there. And soon there will be other things like user cloning. You can build your own templates. You can automate things. You can integrate it with the rest of your kind of computational tools. If you're a tinker, you can use it as a way to contribute, to learn web development. There will be soon support to represent strains and cell lines. And also in the particular for Reclone, I think we could make it quite easy to be able to retrieve sequences from the Reclone kits directly from OpenCloning. So you don't have to be like loading your files or looking through your computer and all this kind of stuff. So with that, thanks very much. This is the people in my lab, the people who gave me the money to do this work and so on. And yeah, thank you for your attention and thanks for the invitation.
Thank you, Manu. This is very exciting. Definitely something at least I would have welcomed during my early researching cloning days. I can see there's a couple of questions in chat. Ben, would you like to voice your question? Or
Sure. Can you hear me?
Yep.
Yeah.
Great. I was just interested in the idea of being able to look through genomes for particular sequences. And something that I'm interested in is taking, a gene of interest and then blasting it into another genome to look for a homolog, and then quickly just grabbing that sequence. And for anyone who's used GenBank, that's not always intuitive to do. So I'm curious if your tool could do something like that.
No, no. So if you run a blast search on your site, it would be easy then to use OpenCloning to load that particular results that you've got. But I don't think you will be able to do it in OpenCloning, at least anytime soon. Yeah.
Okay, thanks.
And Felipe, are you happy to voice your question? If not,
I can
feel free.
I can read it
Yes.
there. He's asking if you can annotate manually. So yeah, this is something I know it's really annoying and you cannot do it yet. There's good reasons I think for it, but it's coming. So I just have to think a little bit what is the best way to represent that, because if you annotate the sequence, you're changing it. So then kind of the history suddenly becomes not deterministic. So I just have to think what is a good way for capturing this in the model. But yes, hopefully soon enough you should be able to, because I agree that that's super important and it's not possible right now.
And I'll go on to Adrian.
Oh, sorry. I thought that Scott was first.
Sorry, I may have missed it. Apologies. Who wants to go
No.
first? Scott?
No worries. I've got a quick question. I actually have to race off to a medical appointment after this, but I'm the creator of the Open Yeast Collection, a Golden Gate toolkit for engineering yeast. And I'm wondering if it's possible to get your tool. First, I love the fact that you're doing persistence and that will come out as part of this, because that's something that I've wanted for a long time. But I'm wondering if you can do a multiplexed assembly. So for example, let's say you've got your yeast construct all set up, but you want to try like 5 different promoters. And so rather than do each one individually, you say, these are the promoters I want, and have it spit out the 5 different constructs.
Yes, so that is not done yet, but it's definitely something coming. But yeah, I just haven't done it yet. But I know that a lot of people are doing this kind of like parallel assembly or like, so I think, yeah, should come at some point. Yes.
Yeah. Okay, awesome. Great work. Thank you.
Thanks.
Adrian, to you.
Yes. So I have a couple of questions there. The first one is not really affecting me, but I know that lots of people from open community, sometimes they have bad internet and it doesn't work. Is there any way to load this or run this locally? So that's my first question. And the second question is, I know, and I've done a little bit of work with BioPython, which seems to be a very popular library. Which library did you use? And in case you didn't use BioPython, why didn't you use that one?
Yes, so the first question, can you run it locally? It's yes, but you have to know how to run a web application in your computer. So you can either run it like a dev server or you can use Docker to run it locally. But probably it needs, I mean, depends on the profile of the people who are using it. So, but it's quite easy to do even if you're not like very computationally skilled. It's easy, but if there is somebody who wants to kind of do that, I'm happy to kind of support them in getting it set it up. But it's really easy, but you need to know how to use Docker mostly. Then for BioPython, so the library that I use is called PyDNA. So PyDNA is a library that builds on the BioPython sequence classes and extends them to add things like double-stranded DNA, overhangs, circular DNA. So
Okay.
and yeah, so I'm a maintainer of this library, and I kind of is what I use in the backend for cloning. Yeah.
Okay, so you buy DNA builds on BioPython.
Exactly, yeah.
Okay, yeah, because I mean, there are several libraries there, but it's kind of good idea to kind of stick with one of them that's more solid there, because otherwise, you know, it's very hard. And I guess in the scripting that you said, where you can extend and you can add your own cloning method and stuff like that, you would use this library too? You could use this library, or you just have to make API calls over the web?
Yes, so right now you basically, it's not really an API call, but you have to use the function that is kind of the same one that the API uses when you make a request from the front end, but that's going to change in the short term. So we're making it so that it will be easier to script, and by using directly the methods in PyDNA, you're generating the history in kind of behind the scenes, because PyDNA has very simple methods like cut, and then you pass an enzyme as an argument, and that would give you the products. So the idea is that it also gives you the history as well as as kind of the sequence product, and then you don't have to like use the website to produce these kind of like history trees, and you can more easily do that. Yeah.
Okay, that's great. Thank you so much, and looking forward to this I don't know, enhancements that you just mentioned. Thank you.
Great. And the last question I can see here for now from Johan. Would you like to ask it to Manu directly? And if not, I can read it out. So Johan asks about the intellectual property rights applied to using OpenCloning, and when publishing the results and when it becomes a product and so on and so forth. So have you had any thoughts about IP?
So the web application, well, all the tools like the backend, the front end application, everything is MIT licensed, so you can use it for whatever you want. You can extend it, change it. I have no claim over the IP, so, and I hope to keep it that way. So at least for now, I have academic funding for 2 years to keep working on this. And in the meantime, I'm trying to kind of figure out a way if I can start a company to start making some money with it. But yeah, the idea is to keep it always on MIT license. And there's other companies who are doing something similar. So eLab FTW, which is this electronic notebook I mentioned, it's also kind of completely open source, and they still make money with it. So that's my hope, to kind of find a way to sustain the project being open source in the long term. And yes, I'll share the GitHub repository now. Thank you.
Perfect, thank you. And well, now we will move to our second speaker, also sharing an exciting initiative and exciting opportunity for our community. So joining us, we have Bijoy Desai. He is a life science professional with over 9 years of experience in end-to-end discovery and development in biotech, biopharma, and diagnostics. He holds a PhD in biochemistry from the University of Illinois Urbana-Champaign, and his expertise encompasses synthetic biology, automation, AI, and machine learning, with extensive experience and leadership in biologics discovery from early discovery to preclinical stages. He's currently the technical project manager at the Align Foundation. Align Foundation is a nonprofit organization that works to propel biology into a new area of reproducible, scalable, and shareable research. Their vision is to accelerate the field by developing powerful predictive models. As part of their community benchmarking initiative, they host the Protein Engineering Tournaments, which is an initiative we'll hear about today and hope to have our community joining. So the stage is all yours, Bijoy.
Thanks for that introduction, Sibel. I am going to share my slides. Just a second. Can everyone see my slide?
Yep, all good.
Great. So, hi everyone, I'm Bijoy Desai, and I will be talking about our upcoming 2025 protein engineering tournament. But before that, I would like to start with a little bit of a introduction. Just to give you a sense of like what is currently missing in the field of integrating AI into biology and biological engineering. Just to give you a sense of where we have come from, the field of structure prediction, it went from not being able to predict structures very well to AlphaFold 2, which was the main breakthrough, and it's shown here by the different CAS competitions that happened throughout the year. A score of global distance test score of 90 is what is roughly considered equivalent to experimentally determined structure. And none of the top participants were able to break through that score until AlphaFold came along, which still did not get a score of 90. And then AlphaFold 2 came along, which came close to solving that problem in CAS 14. So that kind of gives an idea of kind of the task cut out for us. How do we create the next AlphaFold, which would be a predictive as well as generative model for biology, ideally biological function? So then that makes you ask the question, what really fueled this breakthrough? One was clearly a common alignment in the community saying that we need, structures are important and structures help us understand something about function. So that was the key that led to a large amount of funding and led to creation of the Protein Data Bank, a large repository of high quality structures. And then what also was key was an open source community benchmark that kind of kept track of predictive models. So we have experimentally determined structures, but what would be really great is if we can, without doing experiments, predict the structure. And CAS, critical assessment of structure prediction, was an open and transparent benchmark that kept track of what the progress in that field of predictive structure modeling was. And so that's what brings me to Align and our mission. So the Align Foundation, its aim is to accelerate scientific research by improving access to useful research methods and data. We were founded in 2021. We are philanthropically funded, and we have over 150 external collaborators, in addition to about 10 team members, which we are globally dispersed. And we have reviewed and published 16 proposals in 2024, and we have developed workshops, which a large number of scientists and collaborators have attended. And that is basically what we are trying to solve. We have like three main functions. One is to identify what is the next grand challenge in terms of predictive and generative biology. And then we do that by our road mapping initiatives, by generating RFPs and holding workshops. Once we have sort of converged on what is the next grand challenge, the next question is, how do we generate that database, which is equal? What is the next PDB and how do we generate it? So that is our data sets function or initiative. And then we do this by identifying automation partners and collecting large scale data sets. And then finally, how do we test the utility of that data set? And we do that by holding community benchmarks, which not only shows how useful is that data, but also shows where the field is in terms of predictive and generative models. So that in general is what we do, and the different sets of activities. I primarily will focus on the community benchmark aspect, particularly community benchmarking of protein engineering models. So that brings me to our protein engineering tournament. And what makes our tournaments unique is that we are transparent and open, very much to our mission. We want our data to be open source and open to the community for advancement of the community. We provide experimental high quality training data. Like overarching theme of our protein engineering tournaments is that we want it to be as democratic as possible and with as many different diverse set of participants globally. So we provide the training data to keep the playing field level. We also provide compute, again, in the spirit of keeping things level. We provide compute resources to participants. And then we test the participant-generated sequences. So we have a transparent side-by-side testing, as well as that is also like a very resource intensive process. So we try to equalize that aspect by testing the participant-generated sequences. And then we, of course, publish all of the results from the benchmark to the communities to have a degree of openness, and then a degree of critical assessment at the end of each tournament. So overall, our tournaments are divided into a predictive and generative phase. Again, in the spirit of sharing, we provide optional training data for both sets of phases, and then evaluate both of them to generate leaderboards. And then, of course, we publish the results. So these are this is an example from our pilot tournament, all the different teams that competed and all the various different modeling methods that they used. Just to summarize that, these are the results from the pilot tournament, where we asked the participants to generate enhanced amylases, which have strong industrial application. There are specific activities shown here, and the different team performances. The black line is the specific activity of the wild type enzyme. So there were several teams that outperformed the wild type enzyme, but not just for activity. We also asked them to maintain 90% of expression and thermostability for these enzymes. So these are very industrially relevant properties that they were asked to improve upon. And then you can see that I've summarized all the different kind of modeling methods that we use, that we were able to benchmark side by side. So including protein language models, GNN, RNNs, for both predicting and generating new sequences. We in total tested 1,200 sequences that were submitted by participants. Majority of them were novel sequences. Yeah, so we hope to capitulate this and expand this in our upcoming tournament, which I'm going to talk about briefly. The target for the upcoming tournament is PETase, or PET hydrolase. PET hydrolase is an enzyme that hydrolyzes PET, which is a form of plastic, into its monomers, ethylene glycol and TPA, or terephthalic acid. Just to give an idea of the impact of this, everyone is very much aware how much of a problem plastic waste is. In 2024, the plastic production hit 410 million metric tons, and which it is projected to triple by 2060. And then most of that plastic is not recycled. So only 10% of it is truly recycled. Most of it ends up in landfills or is incinerated, creating very adverse environmental and health effects with BPAs, leaching, as well as microplastics in our water and land resources. One of the efficient ways and green ways and sustainable ways to degrade plastic is to use enzymatic means and then degrade it into its monomers. And not just do that, also engineer enzymes that can remake plastic in that way, like truly recycling the plastic and creating like a circular plastic economy. And that has been a goal for a while, but there are key issues that remain for large-scale industrial application. And some of the key issues are high activity of the petases at high temperatures, the glass crystallization temperature that ideally the enzyme should work at industrial conditions. The other important issue is high degree of expression. If you wanted to make several kilogram scale of this enzyme per year, they need to be expressed at an industrial scale. So this year's tournament, we plan to benchmark PETA's design models for these kind of industrially relevant properties. And just to give you an understanding of the structure of our tournament, we are again going to divide our tournament into predictive as well as generative phases. In the predictive phase, participants predict properties of petases. And then in the generative phase, they generate novel petase sequences with enhanced properties. The predictive phase is further divided into a zero shot and a supervised track. In the zero shot, the participants will not receive any training data, and they will be asked to predict petase properties. And then later on in the supervised track, the participants will be provided training data that they can use to train their models for predicting petase properties. And then later on, the teams that do well will automatically be invited to participate in the generative round. And there they'll be provided training data in round one. And then with the help of that, they can generate novel petase sequences. And in round two, they will be provided additional training data, but then they will also be provided the results of round one, and they could use all of that data to further improve upon their sequences, almost like a DBT cycle of enzyme engineering. Just to give you a sense of like what kind of training data we will be providing. So the participants will be provided 6 times 10 raised to 3 single mutational scans of several petase enzymes, but they will also be provided close to 300 natural homologs in the lipase, cutinase, polyesterase family of hydrolases, where mainly petase activity is found. So they'll have all of this diverse sequence data that they can use to train their models, both for predictive as well as generative models for, and then the data will be multi-parametric. So it'll be activity, some form of a measure of thermostability, as well as expression. And then before the beginning of round 2, they'll be provided combinatorial mutant data. So this would be single mutant data and natural homologs. And then right before round 2, they'll be provided combinatorial mutant data, just like this one was single mutant data. And then, of course, they will also have data of their own generated sequences from round 1. So that basically will consist of training data set that the participants will be provided. In terms of how the performance will be evaluated, for the predictive round, it would be either some form of correlative measure, like experimental correlation, or some sort of ranking measure of outputting best sequences. And in terms of evaluation of the generative round, we will be evaluating based on fold improvement or an industry benchmark threshold, while also maintaining activity after heat shock, as well as maintaining basal activity or greater than or equal to 90% expression relative to the industry benchmark. A kind of a sub-award that we also plan to award is how diverse sequences these generative models can make. So there will be some degree of diversity metric that will be used for awarding a sub-award during the generative round as well. So why should people participate? Of course, people should participate for recognition that their model is the best performing. So recognition on the leaderboard. But we also this year have a cash prize that totals up to $30,000. And then, of course, we provide free DNA synthesis and free testing of their protein sequences. We also plan to publish the results of this year's tournament in a retrospective special issue of proteins. So the winning teams have the opportunity to be featured in the journal. And then we also plan to invite the participants to give a talk at a conference discussing their modeling strategy and approach. Just to give a sense of the timeline, the registration for the tournament is already open, and it ends. The deadline for registration is October 17th, with the predictive phase beginning on November 3rd, and then the generative phase beginning on January 5th of 2026. Yeah, that is, I mean, I'm happy to answer any questions, both regarding the pilot tournament as well as the upcoming tournament. So the floor is open.
Amazing. Thank you so much for sharing. It's a very exciting opportunity. As I mentioned, I do hope we have Recon community members joining. Thank you so much for sharing. And we have questions on the chat. Ben, I saw your question. Would you like to ask? Yeah.
Sure. I got a couple questions. This is really great. I've been a big fan of Align for a while. I attended one of the workshops a few years ago, but So I guess the first question was from the 2023 results slide, and I was just curious if those were experimental measurements or predictions.
Sorry, I had a little bit of feedback. What is the question?
No problem. The slide earlier on in your deck about the 2023 results. I'm curious if those were, that one, if those are measurements or predictions.
Oh, that's a great question. Yeah, these are measurements. So these are participant-generated sequences. So sorry, I didn't clarify that. These are results from the generative round. So these are experimental results of the participant-generated sequences. Yeah.
Okay, nice.
And you had a few more questions. Would you want to follow up on them, or shall we intersperse them with Adrian?
Well, Adrian's next, and then I can ask my other questions.
Sure.
Well, actually, I had the same question as Ben, making sure, trying to understand if these were tested in a wet lab or just computational assessment of the results. By the way, it's a great program and a great initiative. Congratulations for that.
Thank you. Yeah. So just to add to that, the results are now have been published in Proteins. So you can go and read more in detail about the pilot tournament. So these are this particular slide is experimental tested participant generated sequences. So they gave the participants submitted about 100 to 200 sequences, and they were all tested. And then you see sort of like the all of the sequences from each of the teams. But then before this generative round, they also were asked to predict properties. So, and then those results also you can find in this publication. So if you search for you should be able to see the results.
Perfect. Yes, I think we've just posted that in chat. So if folks are interested, the paper's over there. And I guess I'll pass back to Ben.
Thanks. Yeah, I'm curious if there's specific. So for the predictive round, are there specific properties you're looking for predictions on, or is it more just like people throw what they have at your tournament and that's it?
Yeah, so we will be looking. So there is more structure to it. So we will be looking for property predictions of three main properties. One would be activity at a specified condition, which will be released right before the beginning of the tournament. The second would be residual activity after heat shock or activity at a high temperature. And again, those conditions will be released. Those specific conditions, including like buffer and stuff like that, will be released. And then the third would be expression in a particular host organism. And then, yeah, those are the three properties that the participant will be asked to predict, given a particular amino acid sequence of the protein.
So
there will be like a test or validation data set that will be withheld, and all three properties for those sequences will be what the participants will be evaluated on.
Okay, so we're given the sequence then, and then we would have to make predictions based on that sequence. We're not generating sequences and making predictions of those properties of those sequences.
Correct. In the subsequent generative phases where you will be asked to generate new sequences with enhanced as improved properties as possible. So there you kind of push the gas pedal and kind of generate as many improved sequences as possible. I mean, each participant will be allotted like a certain set of sequences that we'll test for equal set of sequences we'll test for each participant. But that would be in the generative phase.
Thanks. So my last question was about how to register for the predictive versus generative phases, because the website was just a little unclear about that.
Yeah, so that's a great question. So one thing, so we are a little bit sort of, the generative phase is of course resource intensive. So we might have to cap the number of participants. So a sure shot way to automatically participate in both phases would be to compete in the predictive phase, particularly the supervised part, because the zero shot is kind of optional for every participant. And then if you are the top participant, you automatically are invited. But then we also have applications. We understand that certain labs, they specialize only in generative kind of models. And so we have an application-driven process as well to participate directly in the generative phase. And then based on the uniqueness of your approach, and different factors like that, we will select a limited number of participants to directly participate in the generative phase as well. So those are that's like the general process of how we will be selecting participants to compete in the generative phase.
I did think of another question because there's no one else, so I'll keep going. I'm curious,
Sure.
The website also mentioned sponsors. Could you talk a little bit more about what you're looking for with sponsorship?
Yeah, so I mean, the sponsorship is mostly in service of our main goals, is to make things open, make things kind of democratic, and so sponsors that can either provide resources so that we can test more number of sequences, so more participants can participate in the generative round. So like DNA synthesis or just experimental testing based resources. We of course want to provide unlimited compute, or not unlimited, but as much compute as possible. So any sponsors that can help us provide compute to the participants. We already have sponsorship from certain places that enable us to do that. We would want to expand that both either in this tournament but also subsequent tournaments. So those are the kind of sponsors we're looking for, just to make it more open and democratic.
And increasing capacity by the sounds of it.
Absolutely, yeah.
Okay, cool. Thanks.
Yeah, so if you know anyone, yeah, please feel free to reach out to me, and I would love to have a more detailed conversation about it.
Great.
Amazing. Thank you so much for the questions. We also have another question on the chat from Luis. I don't know, Luis, if you want to ask the question. No problem.
Oh,
okay.
Is the question,
No
would
worries.
the sequences be available freely? Is that the question?
Yes, exactly.
Yeah,
I'll read it out loud for. Oh.
Sure. Sorry, go ahead, Isabel. I didn't want.
No worries, delays, internet, Zoom, everything. So the question is, would the sequences be available freely, both for research and commercial use?
Yeah, I think that's a great question. So as such, the participants are the sequences that they generate, as well as like the modeling methods that they use, or the novel methods that they have invented as a part of this course of this tournament, or even before the tournament. So the participants retain intellectual property on their sequences as well as their methods. We do make the results, including sequences as well as method abstracts, openly available, but there is a substantial amount of time before, from the time of the tournament commencing till when the results are published, which, like, in that meantime, the participants should be able to file for provisional patents or whatever they want to do with their methods as well as sequences. But after that, like, we plan to publish all the results. I hope that answers the question. I mean, one other thing I also forgot to mention is that so the participants will be participating anonymously throughout the competition. So we'll be anonymizing all the names, all the team names, as well as affiliations. Only the top participants will be automatically de-anonymized at the end of the competition. So, but everyone else will be anonymized, and that information will be safely held only with the line, and we will not disclose that information, just to sort of encourage participation.
Oh, amazing. Thank you so much for sharing. Luis also sent their thank you on the chat. So we were talking earlier about the results from the pilot program. Yankee shared the link from the publication for anyone interested in checking. Also on our forum, we have the links about the tournament this year. And as I already mentioned with Bijoy, we'll be sharing about this opportunity throughout Recon social media, because it's a very interesting, a very exciting opportunity for our community. Also with our first talk with OpenCloning. So we're always very excited to share opportunities for everyone beyond this meeting for our global community. And well, so now we have some time for more questions. Can be about the protein tournament for Bijoy, about Alliance work, or about the OpenCloning to Manu. So anyone have any additional questions?
Thank you. I just want to say thank you to Sibel for this opportunity, and thank you all for listening.
Amazing. Oh, thank you. Yes, we have more thank yous on the chat. I think all questions then might have been asked, but as we always say, we have our forum where our speakers are also joining us, where you can send any questions, any thoughts, suggestions you might have. We always have a topic for our community meetings, or you can just post whatever you have on your mind on our forum, be it troubleshooting protocols, exciting new initiatives in open biotech, and everything else. So thank you so much, everyone, for joining us. Yes, Yankee just shared the link to our forum. We hope to see you next month for our August community call. Also, again, with 2 speakers, we're feeling very popular right now, which is very exciting. So thank you really for always being with us. Thank you to our speakers, Manuel and Bijoy. Have a great rest of your weeks and of your winter or summer holidays. Bye-bye.
Thanks again. Have a lovely day.
Thanks. Bye.